You've probably heard about these crazy stories of grandmother cells found in grandchildren or about fetal cells found in some organs of the mother. This is called microchimérism, and it's a really special biological phenomenon. To answer all my questions, my guests today are Amy Boddy and Thomas Kroneis. After a few months of exchange, we finally met at the ESRI conference in the Netherlands. Special thanks to Michael Ekmans that made these interviews possible. So today we will see how this cell travel, what type of cell actually can travel, and what do they do. I discovered Amy and Tom's work in this book, Les cellules buissonnières, that I highly recommend. I think it will be available in English very soon. My name is Alex. I am a fertility awareness coach and the founder of Magic Ovaries. The best way to support this channel is to subscribe or leave a thumb up. Enjoy the interview.
So it all starts on a monthly regime where a group of oocytes, like egg cells, start to grow. And within this group of oocytes, there is one that is particularly good in growing. And this kind of gets really big, right? So there is a fluid-filled area then that is very close to the surface of the ovary, and on a certain point in time this is going to rupture. So the super interesting thing there already is that the tube that needs to catch the egg cell knows actually where this will happen. So there is some movement of the tube towards this point. So once this has happened, the oocyte is starting traveling towards the uterus. It is still surrounded by some cells, the corona cells, that are important to feed the oocyte. So, and on its way down to the uterus, sperm cells are already approaching. So most of the sperm cells kind of wait, get lost, stick around on the way up to the uterine, and just a little fraction, kind of, I don't know, 10, 100 cells, sperm cells, kind of reach actually the egg. And the first sperm that reaches the egg cell, first of all, enters the egg. And second, at the same time, there is a reaction that goes around the whole egg cell then, and that is to shield off other sperms, so that there can be not a second, a third, and so on sperm entering, because that would cause issues. At the beginning then, or the next step actually, is that the sperm nucleus and the egg nucleus kind of fuse, and then the divisions start. So it's important to know that the size of the egg, of the fertilized egg, stays the same, because this shield, this zona pellucida, is very rigid. So until close to entering to the uterus, the cells start dividing, first in 2 cells, then there's a 4-cell stage, 16-cell stage, and so on, until it reaches a stage that is called morula, so where all the cells looks like a berry, right? So with no space within the cells. However, then there's the first cell fate decision we are taking in our life, meaning the cells that are the outer cells will end up being what we then call the placenta, so the organ that connects the fetus after implantation with mom. And the other cells kind of group to what we call the epiblast, and this is going to be then, this is going to be us. So actually, we are more than us, right? We are the cells that are going to be us, and the cells that are then going to function as this interface tissue placenta. So at that point of stage, we are already reaching the uterus. And then there needs to happen the hatching of this now called blastocyst stage. So because the zona pellucida is shielding off everything, so this needs to burst open, and then the blastocyst finds its way to the mucosa, so to the surface of the uterus, and then implantation starts. And this takes a couple of hours to days to get directly into the wall of the uterus, and then we start growing, kind of. So that would be the first 6 to 8 days of our life.
And when it gets free from the zona pellucida, is it already very close to the endometrium? Like, where does it happen? Does it have a long way? This is just a question that comes up, but does it have a long way then to reach the mucosa?
This is a very good question that we actually cannot answer.
Okay.
It's probably close to the surface, but it's not necessarily, it doesn't need to be that. We have no idea of the time frame, probably a couple of hours, that this blastocyst is kind of without any protection until it reaches the area where it needs to implant.
Once we're inside the maternal endometrium, there's an exchange, there's a negotiation, I like to think about, between the maternal immune system and the trophoblasts, those specialized cells on the outside, in order to start breaking down the tissue, in order to start developing the placenta, to give room and nutrient exchange for crosstalk between the mother and the baby. One thing I want to point out during this whole process, what's happening in the endometrium in humans, is we start building up tissue, right? So we build up our, maternal endometrium. We have something called spontaneous decidualization. So we have these very specialized cells that differentiate and prepare for this invasive process to happen. It's highly invasive, and so we've co-evolved this mechanism to prepare for this. And then obviously if fertilization doesn't happen, we remove this tissue, and that's when we menstruate, right? Not all species do this. So humans and a few other, along with primates, a few other rodents and bats actually have menstruate. The rest of the species wait until fertilization to create this really buffy uterine lining in order to invade. So once this happens, we're considered placental mammals, and placentas degree on their invasiveness. And so we are hemicorial placenta mammals, so our placenta is very invasive. And so what happens is there's this negotiation between the maternal immune system. There's very specialized uterine immune cells, such as uterine NK cells, that are cross-talking, and the trophoblasts invade the tissue and start remodeling the maternal spiral arteries. So there's these very nice arteries that line, and they open them up really nicely so that there's nice blood flow and exchange. And you see this growing placenta that can then feed the growing fetus. So it really is this negotiation between, I think, the fetal tissue and the maternal tissue. And sometimes maybe there's the maternal tissue that doesn't allow, potentially, from the maternal immune system standpoint, the fetal tissue to invade far enough, and then it doesn't open these spiral arteries wide enough. And that can be a problem later on in pregnancy, because then there's not enough blood flow going to the growing fetus. And at some point, the fetus says, I need more resources. And the blood flow isn't big enough because the spiral arteries weren't widened enough. And it says, I'm just going to ramp up the maternal blood pressure. And that's a great way to get more resources coming to me. And that's what we consider preeclampsia in the mother, and it can be very dangerous. And there's no cure. The only way is to, like, you know, mediate the blood pressure, but eventually deliver the placenta. Yeah.
Thank you for mentioning the preparation on the maternal side.
Yeah,
I mean, that's super
great.
important. Yeah. So the principal idea of this interface is that you need to secure kind of both sides. Because what happened by now is that some foreign organism is inside or around the other individual. So what you need to make sure is that this is not registered as foreign, and kind of activity started to get rid of it, right? Because it's not the same, it's different. So for a very long period of time, we thought that the placenta, so the interface, is like strictly shielding off baby from mom and vice versa. So as I think everything in biology, there's no 100%. So we learned that there's exchange of antibodies, for example. So mom's antibodies can traffic through the placenta and kind of immunize, no, not immunize, like support the baby with antibodies. On the other hand, baby needs to get rid of metabolites and stuff like that. It needs to intake oxygen. So why babies' red blood cells have a slightly different hemoglobin, binding better the oxygen, so which creates a gradient from mom to baby, so that it assures that, you know, oxygen can be taken up through the placenta. Nutrition as well. So that, I mean, that's the most important organ. That's a very, very important organ for the fetus to stay alive, because that's where nutrition gets in and garbage gets out, and some support like antibodies and so on. So this was kind of the basics that we learned in recent decades. And then all of a sudden it started to get even weirder, because in addition to these no-brainers, we noticed other things to pass as well. So all of a sudden we found maternal cells in the fetus, or in the newborn, and even later on in life of sons and daughters. But also cells from a pregnancy ended up in moms, and this is not restricted to living offspring. Sometimes even pregnancies go unnoticed and are not going towards even a very midterm stage. So the women don't notice that they had been pregnant, and even then it seems to be the case that cells can traffic to mom. So that's kind of the brief overview, kind of, right? I'm pretty sure you're going to add something, because I most likely forgot about half stuff.
I think we always viewed it as a transfer of mother to baby, right? Like, what is, and think about it even from cells, it makes sense, or the immunization, or the transfer of antibodies. Of course, from an evolutionary standpoint, these babies will survive better if they have immune training from the mother. The mother has all this memory and exposure to infectious diseases, and the environment can transfer that knowledge to train the baby's immune system. And so in one way that makes total sense, that there needs to be this exchange of material. And there's some great studies showing that women that are infected with certain things, those babies survive better if exposed later on, and why now it's very important to vaccinate women while they're pregnant, because you can see the transfer happening and the baby can be protected. So that's great. But then there was a study that showed that, yeah, indeed, cells are coming back. And we knew this because now you can do these, like, instead of doing an amnio where you try and test for fetal genetics, you can just take a blood sample, right? But everyone thought, again, at this interface, there's always, between the placenta and the maternal uterus, there's cells dying all the time, right? Cells turnover. And so there was the trophoblast cells, the placenta cells. Everyone thought they were just apoptosing and dying. And then there's like free-floating fetal DNA circulating in the mother, which is great. You can like measure it in blood. But what they were finding is it's just not DNA. There's cells too. And they seem to be cells that persist. And so looking at a woman who gave birth a decade ago still has cells from a baby she gave birth to decades ago, right? And so that's a big question of why and how these cells persist. Because if you learn anything from Immunology 101, it's that we are really good at recognizing self and non-self. And these cells are half foreign to us. And so why isn't our immune system reacting to these cells? I think that's a major open question. We don't know the answer. And we don't know where the cells are coming from. And yeah, we don't know why they're there. We think there's function, but we don't. We're still trying to tease that apart. We don't know, but I would predict that yes, there is a service that happens. I think there would be, if it was something that was so detrimental to our species, it wouldn't exist, right? Like it would have been really selected for or selected against. But we see this being ubiquitous across species that are mammals, this transfer of cells and this crosstalk, that it gives us clues to suggest that it's actually really important and it's most of the time beneficial for the species. But as anything, I mean, our own cells turn against us when it comes to like cancer or autoimmune diseases. As anything in biology, it can get turned against us as well. Yeah.
The fact that we find these then so-called microchimeric cells in the context of certain diseases and so on is not yet telling us whether they are fighting the disease or whether they're causing it. Mm-hmm. So what we need to find is the biological meaning of the cells in the context of when they do good and when they cause an issue. So that's yet not clear. So we see that they're there, and it's super interesting that they can repopulate, well, that's probably too strong, but they can produce every cell type you can imagine. Mm-hmm. And this is only true if the cells that transfer do have some kind of stem cell properties. So then they need to have the ability to either become a nerve cell in the brain, but also a cell of the skin or a hormone-producing cell or whatsoever. So that was already shown. We know that they're there, we know where they're coming from, mm-hmm. but there's this black box where we don't know how they travel, when they travel, what is their importance, what are they doing, and a couple of very seminal papers start showing kind of the biology behind that. I personally like the story of maternal cells that travel to the fetal lymph node, which is where cells can present antigens, like tiny bits of parts of our body showing the immune system, hey, this is something you should defeat, or this is something you should kind of not defeat, more or less. So the idea behind that is that mom cell go to the site where the fetal can start building up immunity against something. So that's like a sneak preview of the outside world, right? So there's obviously a mechanism that helps the baby that has not yet seen something from the outside world to prepare the immune system. So I think that's awesome.
Yeah. And I think if you predict it from the other side, again, we don't know, these fetal cells, we have predictions that they benefit the mother. Like, that's why they exist. And it maybe during pregnancy, they have a different function. Maybe they are helping dampen the immune system, saying like, let me remind you, mother, why I'm here, right? But then why persist postpartum, right? Why? And so maybe they are very similar in, again, helping the mother with her postpartum healing. Could be really important for connecting. I think about it sometimes of like the baby leaving a little bit of an insurance policy behind once they're out in the world, where maybe they're helping with lactation and being able to produce really important hormones that are important in the postpartum period as well.
My question is now, what are we? Because we have these fungi, viruses, bacteria living in us, forming the microbiome. We also have the cells of our sibling, grandmother, mother. So how much can our immune system tolerate cells from other things? And what are we in the end?
Used to try and define self as like, what does our immune system react to, right? Like, our immune system's there to clear it, like create this like protection of some kind of entity, right? And when it reacts to it, then that must not be self, right? Because it's trained so special, like to make sure that it protects the self tissue and gets rid of anything that is not self. But then we learn, oh, there's a microbiome, and oh, that's special, that's fine, they're just bacteria, but oh, there's these other cells, and we starting to question, or oh, there's autoimmune diseases. Like, does that mean you're less, like, And so there's a really interesting work group that I was working with, and they call it paradox of the organism. And it's exactly that, where we're always trying to define what an organism is, but you really need to think about the time and the day and the composition of the organism. And it is a messy collection of different genetic entities, and it changes over time. And your immune system is really great at adapting to it. And sometimes it isn't, and that's when you can see disease and pathology. But it is a definition that continues to change across an organism's lifetime of what is self when you think at the genetic level.
That brings a kind of fluid.
Yes, it's very fluid. Yeah,
into my mind.
Yeah. And we didn't even talk about, we're just talking about the mother-child exchange, but actually we may not have all been pregnant, but we've all been a baby before. And so we have our ancestors' cells in us as well. And so it is this messy collection that changes based on, how many older siblings you have, and how many pregnancies you have, and all the various things. So it's always changing.
Yeah, true. I think it's in part depending on how you define this question. If it's like us sitting here, then I can, you're the individual, I'm another one, and you're the third one. When we now take into account that there is a couple of bacteria and viruses in us, then that kind of expands our being self towards some symbiotic ideas, taking into account that we harbor cells from our parents, from our mother, plus probably also from the kids. Mm-hmm. Then you might think of us spanning 3 generations, right? So what actually is this telling about us? Immune system obviously can deal with these 3 generations. So genetically, we are obviously, in parts at least, 3 generations. So it's very hard to define self. So what question do we want to answer if one of the answers should be, this is self, this is non-self? So I think there are different flavors of self, and you need to adapt the answer to the question. How many people are in here? Yeah, it's 3 individuals. Yeah, how many generations do we see? Probably 8. Yeah, depending who has kids and so on, right?
Yeah.
I mean,
Yes.
that's, you know, several levels of truth at the same time.
Yeah.
I like this idea.
I do too. I love the idea too of like thinking about cells crossing generations too, because usually you imagine, you know, when someone passes, that's it. But actually it's a little more bizarre to think about if grandma cells are still inside you. We usually think genetic lineages end, but yeah, they can still persist.
Thinking about this, so we recently discussed whether knowing that you have cells of your baby still inside would help grieving moms that just lost their child, right? So could that be an information that allows you to kind of cope better, cope faster, cope as such with the situation, or would it rather harm you? Would you don't like to know that probably cells are still in your body? I think there's a lot of this rather, how would you say, philosophical questions that we should address at least in parallel, if not, you know, yeah, in parallel, at least in parallel, I'd say.
Beside their research in microchimérism, Amy and Tom are having another weird research that is about sperm cells crossing embryo during the moment of implantation. In fact, when the blastocyst arrives in the uterine cavity, it has this shield around it that is called the zona pellucida. And in order to go and implant itself to the endometrium, it has to hatch from this shield. And at this moment, some sperm cells can cross the actual blastocysts. Why?
We've been contacted by our Indian collaborators, Deb and Jay, on this occasion because they had quite old data from monkeys where they washed the uterus tract, to obtain blastocysts. What they then found was that in these blastocysts that already hatched from this shielding envelope and have not yet been implanted, they could collect a couple of them, and when they did electron microscopy, so they prepared them and sectioned them in really, really thin slices, and what they could then detect was that these blastocysts had been attacked by sperm cells. So that was actually mentioned like 50 years ago once in a textbook, and has never been seen in the quality that they showed it to us. And they were asking whether we could make up some ideas around that. And this is when we actually got engaged in this research. And we then thought about what could be the evolutionary mechanism that would allow sperm cells to attack a blastocyst. Although I need to say, I think sperm cells attack everything they see. So it's nothing special that they directly go in there. If they're there and can attack, they just go for it. Whatever tissue they see, they're going to attack or invade. So that was how we got in contact with our partners, and then we developed a couple of ideas on the meaning of this, and something is related to that could, it's better you explain, I guess, because it's about evolution and stuff.
Yeah.
Other things could relate then to microchimerism. So what if the sperm cell then stays in there and the developing embryo, kind of takes this information and includes it? So would that be a kind of weird form of, or early form of microchimerism? However, I need to say that the sperm only got into these trophoblast cells, and we know already that it's not us, it's the placenta that is actually being attacked. So that was one of the ideas. So that what if they go, further? Do they also end up in the cells that give rise to the monkey then? Who was that actually? Yeah, what kind of sperm was attacking the blastocyst? Was it the same that was, so the same monkey that was responsible for fertilizing the egg cell, or was it a rival? But that's the point where I would like just to hand over, because that's kind of, could be an evolutionary mechanism.
There is sperm competition in species where, and there's various ways within sperm competition across species, the female can adapt and things like this. But this led to obviously a question on whether this was sperm competition, and it's a little hard to answer because we don't know if the sperm was from the same individual or not. But if it wasn't, you could imagine that it would be like after an individual mates with a female and another mating happens from a different individual comes along, that the sperm would actually be there to maybe block implantation, right, as a competitive thing. And then hopefully next time around in the mating, their sperm would be the one if they mated with the individual again. And so obviously in that idea, it would be a benefit to the competitor to make sure that this female did not get pregnant with the first individual that was mated, but the second, or something like that. I think it also leads to questions on this vulnerability period that there is. Like, so there's this shell where it's protected, but then there's this period where it's not. And if that is indeed something that's really important for the quality of the embryo too. If there is a challenge to the embryo, can it survive the challenge, right? And so is the sperm going to get in and then just cause havoc and disrupt cell division? Or is it properly a trophoblast layer that is actually very protective, and that sperm, there's no problems with it, right? And so it could be also this thing where you see where it's about the quality of the blastocyst as all. And it's definitely, I think, led to some discussions over what is microchimerism, because this is suddenly this, and again, when we go back to like time and space and defining organisms, all of a sudden there's like 3 different genetic entities in this one blastocyst, right? And what would we call this? Would we call this microchimerism? Would we call this, genetic conflict, or what we would actually define this as? And I think this has challenged us. Obviously, a sperm is only has one set of chromosomes, so it's haploid. So it really might just exist as embedded in the blastocyst and not go much farther than that, maybe get enveloped into the trophoblast. But the other idea we actually threw around out there is, what if it is, because there is this period where there's this vulnerability where the blastocyst hatches, what if it's really important for immune challenge? Like it's helping out, actually. Like what we talked earlier about this really important negotiation between the mother's immune system and the developing fetus. And a really good way to do that is to start like showing the maternal immune cells different tags of the fetus saying like, please, tolerate this. And so it could be another mechanism in order to like provide tolerance for the blastocyst. So we went very like, it could be competition, or actually maybe on the other end of things, it could actually be helping the developing embryo. Not everything's competition. Yeah.
Again, it's the good and the bad, you see? So it's tricky to, from such a scarce informational level to, extrapolate and, come up with this and that idea, right? Hard to challenge hypotheses also. I mean, actually, it is kind of part of our work at the very rim, I would say. Yeah, Amy and I co-lead a big project on microchimérism. So what we tried was setting up a project that reconnects the field. Microchimérism research is scattered around the world, and it's part of people doing research in cancer, in autoimmune diseases, so in all these flavors of microchimérism. So what we wanted to do is, first of all, gather this community, get some important people aboard to help us shaping the society and do research on what is the biology of microchimérism. So we want to know, we want to give an answer to, an answer to whether it is helpful or whether it's rather not helpful, or under which conditions, and that is actually more likely to, either or. Rather more likely to say, in this condition, it seems to be helpful. In this condition, it shows up in a not very supportive way for the individual. So the point was that we wanted to get this, the brains together to solve these complex questions. And this is where we do the research in. And in my lab, we are trying to get an idea of how the cells travel. So we are interested in this very first step of the transfer of cells. We're looking into different routes the cells could take and how their environment looks like. So what is the cell type that travels? Where does it travel? And how is the surrounding tissue looking like? This is somewhat different to Amy's work, but you better comment on.
So we're studying pregnancy, and we're studying this crosstalk between the maternal cells and the fetal cells. And so we recruit pregnant women from the local hospital, and we're tracking their microchimérism in terms of the fetal cells going into the maternal blood, but also the maternal cells. We're looking at the cord blood and seeing what's in there, and then also immune phenotyping the women, looking at basically the immune dynamics with the, there's some women, we talk about this as like, oh yeah, there's these cells. They're very rare actually, and they're hard to detect, but some women have very little, and some women have a lot of these baby cells in their body. And we want to really understand the variation there. And so we're tracking these women longitudinally, and we're looking at that variation and mapping their immune components to see if we can start explaining why some women have a lot and some women have very little. Yeah.
We're trying to build up kind of a society. So this was a really, really important first step to get the key opinion leaders together. Now we need to transform to stage 2, where we stay connected, get additional funding, of course, to do research, but also reach out to other people that are in diverse fields of other diseases, but still connected to microchimérism. So I think we need to get the holistic view on the biology of microchimeric cells. Otherwise, we won't be able to solve the riddle on what is the biology behind that and what maybe is the evolutionary effect. So how does this fit together with this super big picture?
As you see, today there are so many questions that cannot be answered, but the research is just fascinating. So thank you so much for watching this video until the end. If you liked it, please subscribe to the channel or share it. I'll see you soon. Bye!